Hi, everybody. We're going to start in about 2 to 3 minutes talking about avoiding fraudulent image manipulation. This is Doug Darnowski, and I will be with all of you in just a few minutes. Um, I will just tell you ahead of time that we'll be taking the last 10 minutes or so to answer people's questions. So here in GoToMeeting, there is your ability to put in a question. You should have some kind of a box over towards the right where you can click on a little arrow and then type in a question. We'll hold those questions until the very end, but we will get to those. So if you have a question that occurs, go ahead and do that. And everybody will start in about a minute or so based on my watch. Okay, everybody, let's go ahead and get started because I've got 8:30 Eastern Daylight Savings Time on my watch. So welcome to the webinar. My name is Doug Darnowski. I am a professor of biology here in Indiana, United States, and I'm presenting this on behalf of Enigo. A little bit about Enigo first, and then we'll talk about fraudulent image manipulation, which is a major problem, and it's something I've personally been aware of in my research work for some decades, and we'll talk about that as we go through. But first, a little bit about Enigo. And a reminder, as I'd said before we got started, that if you do have questions, you can use the little box that pops up here on GoToMeeting, and you can get those questions to me. I'm going to take them at the end. So the last 10 minutes or so of the webinar, we'll switch over to that and we'll take your questions, try to answer those. But feel free to send them in at any time that you want to do so over the computer. So a little bit about Enigo. So Enigo is a company that's been around for a good time, especially in this business. It's been around for a good long time. It's working in a whole bunch of different countries, and their main function is to try to help authors, scientists, to write their papers, understand how to do it best, understand how to avoid pitfalls like making mistakes in producing images, that kind of thing. And they've worked with a lot of people. And so they are a really terrific company. And they have a whole bunch of different services out there. They have Enigo Academy, with all kinds of discussions and materials available. They have webinars like the one that we're in right now to talk about image manipulation. They also have workshops, including ones live at a variety of universities. They have training programs online, videos, things like that, and the ability to consult with scientists, because science can be confusing or sometimes a scary kind of business, and it helps to have somebody to sort of hold your hand through the whole business. They also can do things like editing language for you, which is a big deal. Since English is such an important language for publishing your science these days, they can help you with that because it's not easy. And English can be a confusing language at times. So they also can help with translation and preparation of manuscript. That all goes together, and further help as you're trying to publish your papers, because you want to have your career go well, and Enego can do a lot to help you with that. They can also help you do things like prepare video abstracts and summaries, and with medical writing, which of course includes a lot of technical terms, and it can really help to have somebody around to help you who is really familiar with English, whether it's American English or Australian English or British English, but whatever dialect of English you need for the work that you are doing. So let's talk about image manipulation and what's allowed and what's a good idea and what's a bad idea. And of course, this is important if you're going to present your science any place, because if you're going to present your science, you probably have to show pictures, even if it's just a picture of a graph or something like that. But more likely, you have to show photographs. And there are some things that are good and which you can do to help people reading or viewing your work, whether you're writing a paper or giving a seminar, to better understand it, to see it better. But there are also some things that you can do which are not legitimate, okay? So let's take a look at this. First of all, even before the way that we can manipulate images today using programs like Photoshop or the free ImageJ program, which are originated with the United States National Institutes of Health, and which is still free for download, or various other programs, even before digital manipulation, it was possible for people who were skillful in the darkroom in photography to change images, like the example that you see here, in order to claim that they had scientific results that they really didn't have. And let's face it, there's a lot of pressure. You want to advance in your career, you want to get grants, you want to get jobs, and probably today, with everyone worried about this virus, there's even more pressure. People are even more nervous. And so there's the temptation to falsify results. So here you have an example of from a paper, and this is 1981. So this is before Adobe Photoshop was in existence. And the idea here was the researcher was claiming to have found evidence of a protein kinase cascade. That is, where you're going to be adding, the cells will be adding phosphate to proteins, and that will be detectable used by adding radioactive phosphate, 32P, as you see on the right-hand side of the image, to cells. And basically, when people looked at the paper, they said it doesn't make a lot of sense. And it turned out that this person was just taking molecular weight markers, and if you haven't used those, if you're not a molecular biologist, they're the equivalent of a ruler. So when you are separating proteins and you want to know how big they are, you need a ruler. And you can buy a set of standard size proteins and put them in the technique in the gel you run next to the things you're doing research on, and that they will act as a ruler to help you understand what's going on. So that's what we're worried about in this case, and this person completely falsified the results. I'll mention to you that one sign that images have been manipulated in a bad way is that the images will often look too good. They will be too perfect. And we'll mention other examples as we go through, but if they look too good or too perfect, it often means that someone has cheated, either by doing something like this, where they're simply just taking the proteins that are not the ones they're claiming to work on, or it could be in an image that if you see noise, that is sort of fuzziness in images, and if it's repeated over and over, because that noise cannot in any realistic way be repeated, that says somebody copied things and reused them, and that's illegitimate. We'll see examples of that. But in any case, so that's an example of this. So what can you do? When should you stop? What's allowed? Well, you can see in the green here, what are okay are things where you do simple uniform adjustments. If you do brightness and contrast, if you have ever taken pictures when you took a trip or you meet friends and you adjust them so that they look nicer, you've done brightness and contrast. That's legitimate. And also color balance, same idea. Cropping, that is cutting the picture, as long as what you're doing is simply cropping out extraneous, not needed things, or changing the size so it fits better in the image you're going to publish, or taking several images that need to be in one figure and putting them together. That's all okay. Basically, what all of those things do is they take the data that you have created and they make it easier for someone who hasn't been doing the work to see and understand it. You should never do anything with an image where you make a fundamental change in your data. In other words, where you change the data in some way where people can't tell that, and where some of the data is changed, but not all of the data is changed. This is one reason why later we'll hear that you should never use filters. There are lots of filters, things like sharpening or softening, or you've probably seen these if you've used Photoshop or a similar program. Those are fine to use if you are editing your vacation pictures or pictures from a family dinner. But for science, you don't know what the filter does, and it changes some data and not other data in ways that you can't control. Never use things like that. All right, in the yellow, things that are inappropriate but don't necessarily don't mean that you've committed fraud. You're not lying, but you really shouldn't do it. In the yellow, we have things like your journal maybe says, here's how we want you to treat your images. If you go outside that, that's no good. If you do other things like adding backgrounds, that's not good. Okay, it may not change the result, but if the journal said don't do it and you do it, you can get in trouble. You can hurt your career. And finally, on the right in red, we have the idea of misinterpretation, and this is going to be deliberate. You delete things or add things like in a protein gel, but you can do this also, say, in an image from a microscope. You can manipulate it. Also, some of the things in green, if you did them to cause a misinterpretation, would be completely fraudulent. So for example, let's say you take a picture and there is something in a micrograph or a picture you take in the field or some picture that doesn't fit with your research conclusions, and you crop out the part that disagrees with what you want to say in your paper. That's fraud, that's lying, that's wrong, and it's going to get you in trouble. So you have to be careful about that. Okay, so those three things: acceptable, inappropriate, fraudulent. Alrighty, examples of this kind of thing. This might look confusing, especially if you're not used to looking at either micrographs or gels where we're separating biological molecules, but just notice a few things. Notice that, for example, on the left-hand side, one of the bands in lane 3, so the top left, in the original, completely disappears from the manipulated image. This is top right. Same kinds of things in B on the bottom, except bands, have been added to lane 3. And notice also that the bands in lane 3 on the bottom look exactly like the ones in lane 2. That's what I said earlier about noise. The background, little bits of gray, spots here and there, if those are identical from one part of a figure to another, it's been manipulated. That kind of background noise does not occur more than once in nature. That's always a clear sign that things have been manipulated. And you can see this again, this kind of thing where people are playing around up on the top right of the whole slide where bands are being played around with. Similarly, look at the micrograph in B on the right-hand side. What's happened here? Well, those are plant cells with the cell walls removed, so they're round. We call them protoplasts. And they're doing a kind of microscopy called fluorescence, where you use a stain that when you shine one color of light emits another color of light. And that's a good technique, but what they're doing is claiming to find certain kinds of filaments in the cells. What's a problem here is if you look carefully, they have manipulated the background, or you can see rather the manipulation in the background. So what they've done is turn the brightness up so high that they create the image of things that they were hoping were there, that they need to be there for the paper to be published, but which aren't there. And you can see in the bottom part of, on the right-hand side, B, how bright the background is. That's a sign that they turned up the contrast in that one very, very bright cell, but not in the control on the left. And so they create an artificial difference. That's also cheating, that's lying. There are lots of other examples of this. I remember when I was an undergraduate, so this would be almost 30 years ago, someone published a paper in Nature on the opening and closing of ion channels, and they had recordings of electric current running through cell membranes. In the recordings, they could clearly cheated because what happened was they had the same exact noise in their electric current data in figure after figure after figure. And this paper was published in the journal Nature. It was complete fraud. So we have to be very, very careful about this. And we don't want to do it accidentally either. What's the problem with doing this? Well, first of all, it's lying, okay? You don't want this on your soul. Second of all, number one, it affects research integrity. It hurts the whole field of science. If we can't trust science, then we can't help people with the things we can learn. B, it makes it hard to reproduce what you did, and that's the whole basis of the scientific method. So you're throwing out the scientific method, you're making scientists look bad in other people's eyes. C, it damages not just science as a whole, it damages your work and that of anybody else. So even if you don't manipulate the image, but someone else on a paper, and so many papers have many authors today, if someone else on your paper manipulates the data, you look bad. You're sharing in their lie. So you have to learn to look for signs of manipulation so that you can avoid it if you have a dishonest coworker. In D, if the paper is published, you'll have to retract it, which is a major embarrassment and will damage your career. And in E and F, you may be fired and your career may be over. You may have spent 10 years or more becoming a scientist, and it's all gone because of one bit of cheating. And it might not even be your cheating, but it may be someone else. And if you don't pay attention, you can destroy yourself in that way. So you have to be very, very careful. Even if you're trying to be honest, not everybody in this world is. I think we all know that. So on the digital side, not just going into the darkroom with photographic negatives, how has this been done over time? Well, Photoshop is the main image manipulation program out there, and it has been around now for over 30 years, and for about 30 years, it's been licensed and sold by Adobe, which company produces lots of other digital tools. It allows us to enhance digital images, and it's very useful. Everybody uses Photoshop at some point in their career probably. But because it makes it so easy to do this, people who are unethical, people who want to lie and cheat, can do so much more easily. Okay. Photoshop over time has had more and more tools. Originally, you had color correction, and you could do brightness and contrast, and you could clone things. That meant you could sort of copy a little piece and reproduce it elsewhere, like the bands that we just saw. Other things have been added over time, like various kinds of filters. It's very sophisticated today, and it gets harder and harder for detection to be done. So for example, here's your clone stamp tool. You can see here in this image how the morning glory flowers here that you see, the vine is growing up some sort of a small tree, and how someone has been able to clone the top flower and make a copy of it below. Right. And in fact, notice that they've also cloned the bit of green leaf behind the flower as well. So it's easy for people to do that, and that's how they can make bands that aren't supposed to be there. Or you can clone a blank part of a DNA gel, let's say, or of a microscopic image, and you can cover up something that you don't want to be there. So the clone stamp tool can be a dangerous tool in dishonest hands. How do you avoid this sort of stuff? Because you can make mistakes anyway. Okay, you can make mistakes even if you're trying very hard to be honest, and you don't want to hurt yourself accidentally. First of all, like I said earlier, don't do anything that fundamentally changes the data and can mislead the reader. Be very careful that your original data is there and all of the data is treated in the same way. It's consistently. Also, pay attention to your journal, or if you're not, say, presenting in a journal, but you're going to go to a meeting after this silly virus goes away and everybody stops being afraid, you want to follow the rules set up by whoever is running the journal or the meeting. And also tell your co-authors what you're doing, and feel free to ask your co-authors, what did you do to get the images in the paper that we're going to publish together? And talk about it. If you're concerned, talk about how it could hurt everyone if they're not careful and not honest. And then finally, look very carefully at your own manuscripts. And if you're a peer reviewer for a journal or for some other publication source, make sure you take a close look at the images yourself for the signs of unethical manipulation. There have been a number of milestones over time where as we have recognized more and more how this manipulation can occur, and we don't have time in this webinar to go through these papers, but these are good references to have, and this webinar is being recorded, so you'll be able to go back and look at this slide at some future time and get these and look at these. It'll tell you about what not to do, how to avoid these kinds of problems. Do's and don'ts, and the green ones are things you can do, should do, and the red ones are things that you should not do, you should avoid. Make sure your changes in your images or other data are uniform. Don't change some of it and not other parts of it. Don't use auto adjustment tools. That includes filters, because you don't know what they're doing, and it can overprocess things, and it can create things that are not really there. It can create artifacts. And this is the case if you use filters, where you can accidentally commit fraud and hurt yourself. Filters will add things. So you might run your images through a filter and say, oh, look, I found something that I didn't notice before. Well, it's not really there. The computer added it by accident. Number 3, you can take out parts of images that are extraneous. That is, if there's a blank area and you need to save some space and it has nothing to do with your experiment, you can crop it out. That's a perfectly legitimate thing. I show my students in my upper level undergraduate courses these things every semester about image manipulation. Also, don't crop 2 different images and merge them. What this means, it doesn't mean you take 2 different pictures and create a figure where you're saying, oh, here's part A and part B. That's okay. What it means is, don't take 2 images, glue them together in Photoshop and claim that they're one image, okay? Always make it clear, if you need 2 images, make them parts A and B of a figure. Don't glue them together and claim they're one picture. Also, you should always have scale bars, especially for things like micrographic images. You don't need a scale bar for a gel in the final publication, but having a scale bar, a line that you add to the image, will help you tell what's going on when you resize the images. It can also help you make sure you match them up at the same magnification. And don't try over and over to do this. Now, that doesn't mean that you can't sit there while you're trying to decide on the size that you should make the images in the end, that you can't try different sizes. What it means is, don't do something that's going to make a fundamental change in some parts of your data and not others, so that it becomes fraudulent. All right, what kind of information do we have out there about guidelines and policies? What's allowed by different journals, let's say, okay? Well, Health and Human Services in the United States has led in this. They're the American government department, which covers the National Institutes of Health, which has had a lot to do with this whole question of image manipulation and making sure things are ethical. And so that's why we have them in this slide. What are their guidelines? Well, if we start on the at the blue and then go clockwise, um, remember that images are data. Okay, it's the same as numbers in a table or a numbers on a graph, something like that. Okay, images are subject to the same requirements and restrictions. Always save your originals. Okay, so if your original material is a digital image, which most stuff is today, when you create a new version of it, change the file name. It would be smart to make an archival copy, just a copy on a thumb drive or a hard drive or somewhere in the cloud of the originals where there's no place that you might accidentally change them. Okay? Because the journal may demand to see originals, and if you don't have it, you're in big trouble. Make simple adjustments, brightness, contrast, cropping out things that are not necessary, adding scale bars, adding simple labels. Another tool that's really useful in Photoshop and other programs like ImageJ will have a similar tool is something called levels. Basically, the human eye is able to see approximately 200 shades of gray or shades of any color. And what Photoshop does is it creates a little, under the, as I say, the levels tool, it will show you the level of brightness for all of the pixels, all of the dots in your image, from black to very white, or from, for any color, in 256 channels or levels. Now, why 256, not the 200 I said that the eye can see? Well, if you think about it, with computers, nobody buys a thumb drive or a hard drive or any kind of memory with a computer. You never go and get, say, 17 gigabytes of something. You get 2 or 4 or 8 or 16. This is a series of powers of 2. And so as a result, Photoshop uses the first number in that series, 2, 4, 8, 16, etc., which is at least 200 for the number of channels it uses for shades in the levels tool. So that's in that series, the first number bigger than 200 is 256. It's 2 to the 8th power. And so if you go into that levels tool in Photoshop, you'll see this graph. Well, you can use sliders there to essentially do kind of brightness and contrast at the same time. It will also allow you to crop out channels that are empty. So if you look at the little graph and there's blank space at either end of the graph, you can legitimately move the slider and ask the computer to recalculate the image. And what it then does is it spreads the data back over those 256 channels, all of it. So you're manipulating all data at the same time. And that levels tool is a very useful one, and almost no one pays attention to it, but this is a really important one. If I were you, and if you do this kind of stuff on a regular basis, and you have Photoshop, as soon as we're done, go to the part of Photoshop where you have the brightness and contrast tool, and right next to that in the control panel or in the drop-down menu will be levels. Look at levels, familiarize yourself with it. It's a very useful tool. And you can go back and forth, brightness and contrast and levels, to simply make the image look as clear as possible repeatedly. That's totally legitimate. Next box, that gray one on the right-hand side, cropping is acceptable as long as you're not cropping out inconvenient data, data that goes against what you're saying in your science. In the green box, compare digital images acquired under identical conditions. It's essentially saying make a control so that you can make sure that everything looks like it makes sense. Everything, your camera is working well, and also whoever's giving you the images is actually producing the same amount of background when the microscope set the same way, or the same amount of background when the gel is stained in the same way. And then last, you can do manipulations to complete the image. That is, you can add a scale bar. That'll probably be required by a journal. You can add labels. Photoshop makes it very easy to do that sort of thing, okay? Alrighty, sorry, I just got a reminder. Just moving my webcam on, sorry, my screen. I just got a message from the people from Enego that they wanted me to do this. Let me just resize my webcam, and I'll move it up here. It's the best I can really do. Alrighty. Oops. All right. Some other things. Again, starting with the green in the lower left. Don't use filters. Filters do things you can't control. Don't ever, ever do it. Don't clone objects into an image. Okay. Again, that's changing part of the image and not the whole thing. Measure and calibrate the intensity of digital images. That's like the problems with background that we saw in one of those first slides with protoplasts. Don't irreversibly compress the image. Compressing, even though it's convenient, especially if you don't have enough bandwidth for your internet, compressing also makes irreversible changes. Oh, it can, depending on how you do the compression. Check magnification and resolution. You want to make sure that you have images that are consistent in terms of their magnification and resolution. And check your size in pixels in the image. You want to have the same resolution, which again, you can change in Photoshop or other programs in every image. Okay, so just remember all of these important steps. Okie doke. And we're about halfway through here before we get to our question time at the end. So this is what we're looking for. Don't improvise. That would be you're changing one part, not other parts. Adjust the whole image. Again, be consistent throughout your data. If you're grouping images, make sure it's clear. That's like saying this is part A, part B, part C of a figure. Don't glue them together and make it look as if, or even create the appearance that they're one image. And remember, because if you can't show up with your original data, you got to archive your original digital images. You may get in a lot of trouble. The journal might accept it. Someone has a question. They say, well, show us the originals. If you can't, the paper's gone, and it's a major humiliation and a major damage for your career. So just don't take any chances. Archive your data. And if I were you, I'd archive the data in more than one place. I'd archive it on the cloud, but I'd also stick it on a thumb drive or even a CD or DVD and stick that someplace very safe just to protect your career. Also, make sure you follow the guidelines for whatever journal you're publishing in. And here, for example, they're talking about the biology of reproduction, but there are lots of other journals out there. There are thousands of journals today. Make sure you follow the guidelines. Those are always found in the print edition. They're found online. If you want to make sure you have the right version, email the editor and ask for a current copy, okay? And make sure especially you pay attention to things like digital image integrity or whatever the similar name from a particular journal is. Have your original images available. They may very well ask for them. And if you don't do that, ethical violation damages your career. You don't want that. All right, so what are the journals going to do to catch this stuff? Okay. And again, we're looking at the U.S. Department of Health and Human Services because of the leadership they've had in this area, and they have an Office of Research Integrity. They have tools for doing this sort of thing, finding problems with unethically manipulated images. To allow you to compare images to show direct superimposition. Whoops, sorry, let me go back, whoopsie. To allow them to do forensic analysis, make sure that it hasn't been messed with. To go through a series of images, groups of images, to make sure that there aren't inconsistencies, like the ones we saw in the protoplasts in one of the first slides in this presentation. If you want to go back later, that's slide number. It was slide number 11, if you want to go back and see that. In the bottom right-hand part where you have images of plant protoplasts and fluorescence microscopy, they can compare that automatically and say, oops, the background is different. And then they'll go in and look very carefully, and then there will be trouble for whoever did that. Tools for doing this, okay? A range of them. Okay, actions and droplets. Droplets are kind of like tiny internal controls. If you're familiar with things in web design, they can create single pixels and stick them in a web page that help to report back to web designers or web site owners. You don't notice it's just one pixel, but that pixel is there reporting back. Well, they can create similar kinds of things in images to help them figure out what's going on. So I should have started up with A, but there's the droplets, different kinds. Adobe Bridge is a useful tool. ImageJ also, and ImageJ is essentially a free program, very much like Photoshop. So it has things like brightness and contrast and levels. It may not call the tools the exact same names as in Photoshop, but they are functionally the same. And it also is a very useful tool for research. It lets you do things like, say, count objects in images. It's a very versatile tool, and it's free, unlike Photoshop. And as I say, actions and droplets for Adobe Photoshop. So lots of tools out there they recommend. And again, if you are ever the principal investigator, the PI for a laboratory, you want to take very great pains to watch what your postdocs, graduate students, others are doing with the data. Use these tools and check to make sure someone isn't committing fraud. After all, there's a great temptation. It's not an easy thing in science. You work very, very hard. You work very, very long hours. I remember in graduate school putting in over 100 hours a week at times, and you're paid very, very little, and you want to move on in your career. And if your experiments aren't working right, there's a great temptation to not manipulate your images ethically. It's also a good argument for being very, very kind to anyone who is a subordinate and talking to them and doing everything you can to them to try to be decent and charitable to them. Hear a little bit more about those tools. Again, forensic droplets, excuse me, useful for, again, for comparing things like backgrounds, like we said. Adobe Bridge, where you've got multiple images and you want to compare things like backgrounds, but other features as well. ImageJ, especially for doing things like gel bands, because it allows you to quantitate what you see, quantitate darkness and things like that, and also make comparisons. And then finally, Photoshop has some of these same features as well. The publishers will have these guidelines, and this is not an exhaustive list. And after all, most of us don't publish a lot of the time in things like Cell or Proceedings of the National Academy of Sciences, but these could still be good guides if you look for, say, Cell Press at the National Academy of Sciences or Rockefeller University Press or Wiley. Even if you are not publishing in that particular journal or a journal covered by one of these organizations, you still might get some useful advice for wherever you are going to publish if your journal doesn't have as clear or exhaustive a list of what you should and shouldn't do. So that's another thing to keep in mind. Other resources out there, a variety of ones. Again, this list, this is slide number, oopsie. This is slide number 29, if you want to go back after and are listening to the recording later, and you can go ahead and look these guys up online. Best practices in handling images. There are consulting firms, just like Enigo is out there, that can help you with your English and help you with all aspects of your paper and do a great job for you. In the same way, there are companies out there that can help you do this. If you're a principal investigator, or if you have some concerns about co-workers or collaborators, they can help you make sure that your collaborators aren't cheating in the part of the research they're doing. They can also help you check out your own people, and they could even take a look at your images and make sure there's nothing suspicious, even just accidentally, so you don't, again, hurt yourself by accident. And there are plenty of cases of this, including very recent ones of digital manipulation that's been unethical. So, for example, Boston University in Massachusetts in the United States, they looked at a full professor of medicine in their pulmonary center in their School of Medicine, and they found he'd done a whole bunch of things wrong. You can go online and you can look up this specific case They have it laid out in more detail than we have here on this slide. But in any case, he was given a very significant punishment, 5 years of being tossed out of being able to get grants and to do other kinds of scientific work, because his falsification of data was pretty widespread through a bunch of papers and presentations and grant applications to the National Cancer Institute in the United States and the US National Institutes of Health. This sort of thing has happened lots before. Brigham and Women's Hospital, there on top, that's a big research hospital in the United States in Boston, Massachusetts. They had to pay back $10 million because of fraud in cardiac studies. Science, most of us would give our eye teeth, as we say, to get a paper into science. And there's been plenty of fraud there. And I mentioned to you a paper I saw 30 years ago when I was an undergraduate in Nature. Published, no one noticed, none of the reviewers did. Completely fraudulent work on ion channels, currents moving in and out of cells, so on and so forth. And there is another one listed in Nature, a fraudulent paper. And we've probably heard of other examples like this. So fraud is out there. There are oftentimes signs in a paper that you should be suspicious, not just the images themselves, even before you get there. If you have a very large number of authors for the kind of paper that it is. Now, I've seen or heard of physics papers with over 100 authors, but for a particular field, that might not be unexpected. But if you say way more authors than you would expect, you should be suspicious. If there's a lot of inconsistency in the style from one figure to another, or if you see some of the obvious signs we've talked about, the backgrounds are very different, the background in different microscopic images, very different, that should make you be very suspicious and want to take a look very carefully. And journals are very cautious, especially in a day and age where you can be sued, especially in the United States, unfortunately, where you can be sued. People are very careful about refusing to publish things, but they put an editor's note in this paper. It had to be retracted. So you got to be very, very careful. Look for not only obvious problems with the images, but if other things about the paper smell bad, there's probably a problem, okay? So this is showing you notice this very large number of authors, etc. And again, this thing got into Nature, had to be retracted. And there have been other cases like this. And there are other things too. You think through the science, and if the science doesn't make any sense, don't trust the paper. There was a paper published years ago for political purposes it was either Nature or Science that had to eventually be withdrawn, claiming that there was proof that the American president Thomas Jefferson had fathered children by a slave of his. They used techniques which were completely illegitimate, and it was basically at the time intended politically to help the current president of the White House, who was in the middle of a scandal. Not mean current today. I mean about this happened back in the 1990s. But the paper had to be retracted a few years later. It was done for political purposes. It's sad that this is the case, but we have to be more and more careful today in science. And so here you have an image on a paper, and notice here some things that we can pick up on. First of all, this particular figure is full of all kinds of crazy things. So for example, notice the green four-sided boxes, okay? Notice that the bands look very similar, but when you first glance at it, you say, well, they're not exactly the same. But then you pay close attention to the shape of that green box, you'll notice someone has simply taken the bands that are under the label BN.3, flipped it 180 degrees, mirrored it, and stuck it into the next part of the paper. Similarly, if you look at this band here that I'm circling with my pointer, I think that's a gamma PVEC, and 2-H11, it's the same image. What they simply did was they took the image from 1 gamma PVEC and compressed it, I'd say about 30%, and produced this other image. And that's a little harder to pick up because you look at it and it looks to me also as if it's been compressed a bit horizontally and offset to make it harder to notice that that's the case. And there's lots of other examples of this in the paper. Notice also, the easy way to pick up on this is the fact that you have identical bits of background noise in all of the lanes of this gel, which have been circled with the same color and same shape. Background noise does not occur over and over again. It just simply doesn't do that. What happens is when you see that identically, that says people have been manipulating the data. Alrighty, so that is our last slide. And so now I am going to open up the questions and see what we have in the way of questions. We'll take maybe 10 minutes for that. And then the good folks from Enigo, after I leave this webinar, I'm going to leave, but I'm going to leave the webinar on so that the good folks from Enigo can come on and talk to you about some of their services. Okay. First of all, we have someone simply saying thank you, and you're very welcome. It's been a pleasure being able to talk to you and today and to share this stuff with you. We also have a question, someone simply saying hello. I guess they were testing the question feature. Good talking to you. It says that person's in New York, USA. I hope you either have lots to do or plenty of bandwidth if you're in New York City and locked down. I'm in Indiana, USA, and I'm limited even though I live out in farm country. I hope you're keeping happy and healthy with everything that's going on. I don't see any other particular questions yet, but we're going to stay on here for a few more minutes and before I take off and the good folks from Enigo come on here to talk to you some more about the services which they offer. And I'll tell you that in my experience with them, they are a friendly and helpful and cheerful bunch of people. Someone else has said thank you for the presentation, and you're more than welcome. I'm glad that you were able to come today. Looks like someone has, well, there's a raise your hand tool that popped up, but it went away all of a sudden, so maybe someone was just testing that. And we've had a good attendance today. There's well over 50 people. And I think the folks from Enigo are starting to interact with you a bit. So if any of you would please be willing to answer this quiz, it'll help them get an idea about how people liked today's webinar. I'm on here for about 5 more minutes if anybody has any questions. Just scrolling down. Oh, wait, here we go. Sorry, got a couple more questions now. The stamp in Photoshop, someone asked, how can you recognize a stamp in Photoshop? And an easy way to do that without even trying to use some of the more sophisticated tools we talked about at the end of the webinar, real simple. Look carefully at very similar images and you're going to see the identical background. Now, if the background looks fairly flat and gray, try playing around with brightness and contrast, because especially if you turn up the contrast and maybe turn down the brightness a bit, pixels will start to pop up more clearly. And if, again, you have the identical kind of background in multiple places in one image, that says that the clone tool has probably been used. For small journal publishers, how do you deal with this sort of thing? My first recommendation is look for what you can get. Look for some of, first of all, the common sense things that you can do. That is, just train your people to look for identical background. That is identical in an image. If you have the same background noise, and it's always there, I don't care how good the image is, you can pick it out by playing around with brightness, contrast, or just looking for it if the image has already got the right brightness and contrast. Also, ImageJ is free, and that's a terrific tool that was on the list from NIH's Office of Research Integrity. That's a free one, and that can do an awful lot to help you there. So I'd go with the free ones and then the common sense tool. Common sense can help you avoid an awful lot of problems in life. And so just use those common sense things. Next question is about axis manipulation in charts. So this is moving away from images a bit, but there, two things, I suppose. One is, and that would be harder than the image stuff because there's not exactly the background, but one is just train people to pay attention to those. We oftentimes just look at images that have been sized the same and assume that the scale or the chart axes are supposed to be the same. Just read them, number one. It might be harder to pick up on because you, in theory, in making a chart in something like, say, Excel, could create a label and paste it over the existing numbers and then fake the axis. That would be kind of like making a clone stamp tool, but in Excel. That's a little trickier. And then the attendee also asked about the stamp tool. That's what I was saying. Use common sense and use things like ImageJ. Someone else from Brazil said, thank you very much, and you're very welcome, and I hope everything's going well for you in Brazil. Beautiful place, and I work on carnivorous plants for my main research, and Brazil is one of the best places in the world for those. Someone else has asked about having the presentation, and Enigo is recording this, and I'm sure that when they talk to you in just a minute, when I'm done with the question time, they will tell you how to get a copy or access the recorded copy. Someone else said, thank you, you're more than welcome. And again, I hope what we've said here could be useful in all kinds of fields because it's not just science that uses pictures of gels and micrographs. All kinds of fields can benefit from paying attention to these things. Someone else has said, thank you very much, and you're more than welcome. Plagiarism from other papers, how to identify that. That's beyond the scope of this webinar, but there are lots of tools out there. Depending your university, there's a decent chance your university may provide this for you. Ask around. You probably have some sort of an ethics office. I know that when we do instruction of undergraduates, my university provides us with free tools where you can upload a copy of their paper and compare it to anything on the web. You may have to sit there and wait a little while the system searches the web and compares text, but you can do that. Another way you can do this, I've found just with student papers, is if you read the manuscript and you find sentences which just don't fit. Okay, in other words, someone's writing and suddenly the writing sounds very different, because usually when a large paper is written by many authors, one person will write the paper, and so the style is similar. If you suddenly find a piece that doesn't fit, copy that, type it into Google, search for it. You can usually catch people that way very easily. I've caught many students cheating that way. I've even caught people in job applications, scientists, plagiarizing that way. Another question. Someone says, if you've got spots of dust, can you remove them using Photoshop? No, it's not, because that's manipulating just one part of the image versus the other. You could try labeling it that way. You could remind yourself just to clean the microscope. I know it's frustrating. I've done lots of microscopy in my life, but that's not a legitimate thing, is using Photoshop to remove dust, because you might remove something else accidentally. Someone else had a little problem. Hopefully that has been fixed now. Try this again. Sorry. Thanks in the United Kingdom. You're more than welcome. Tool good for plant biology images. It's the same kind of thing. The ones that we've listed, and again, if you want a free one, start with ImageJ. It's free out there. And that's about the end of the list of questions that we have. It looks like most people were fairly happy with the webinar today. And they've asked me to go to the thank you slide. Whoops, retraction watch. Oh, and here's with the post query. Sorry, folks, I didn't have this page of those. And there's your thank you slide. So I am going to go ahead and I'm going to check one more time real quick for any more questions. So we don't have any more questions. So I am going to get out of here so that the folks from that the folks from Enigo can talk to you a little bit.
We thank you all for attending today's webinar, and we hope you found the session informative. After the session, as you leave, you will be prompted to fill the survey for the webinar today. Please share your valuable feedback with us to help us improve and so that we know your feedback. Thank you very much for attending today's webinar. With regard to the PowerPoint presentation, unfortunately, we do not share the presentation in a downloadable format. Although we'll be sharing the the link to the webinar recording via email, as well as publishing the recording on our website shortly. Thank you very much for attending today's session. Have a great day ahead.